Hello students, welcome to the theory class of seed production of vegetable, tuber and spices crops. Today's topic is on seed germination test, which is a part of seed testing. To start with, let's discuss some of the important questions. What is seed germination test? Why do we need to go for seed germination test, and when are we going to do seed germination test? So coming to the first one, it is a test to determine the maximum germination potential or viability of the seed. Okay. So germination potential, also known as seed vigor and viability. So basically it means to determine the seed vigor and viability of the particular seed. And why do we need to go for seed germination test? It is because to know the germination rate of a particular seed lot, okay, and to understand how well it will perform in the field, okay, for a particular seed lot. Okay, so we have to determine prior to any sowing or any leveling, so we need to perform seed germination test in order to determine the uh, performance of the seed in the field prior to sowing. Okay, so we don't have to sow all the seeds from the seed lot. We have to take seed sample and we have to test the performance of it. Okay, so when are we going to do for seed germination test? When we have to go for sowing, when we have to go for leveling, and when seed certification is needed, and when it is demanded by Seed Act and seed law enforcement. Coming to the principle of seed germination test, it is usually conducted with pure seed fraction. Basically it means it is conducted with the working sample where the pure seed fraction from the working sample. Okay, we have already discussed the several fractions, several components of the working sample, which are pure seed, then uh, inert matters, wheat seeds, and so and so. From that we are going to grow the seeds from pure seeds only. And uh, the replicates. Okay, coming to the replicates, a minimum of 400 seeds are required. Okay, how much amount of seed is required for testing, how much amount of sample seeds in the sample is required? It is 400 seeds, and it is to be sown in 4 times 4 replicates into 100 seeds. Say if you have a tray, okay, you have to grow in 4 trays, 100 seeds in each trays, okay, which makes 400 seeds. And if you don't have a tray which you can sow as seeds, 100 seeds, if you can sow 50 seeds at a time, so you have to sow in 8 trays. Okay, or in case if you have a smaller size trays, you can which you can sow 25 seeds at a time, so you have to multiply the uh, number of plates to 16 times. So all of this makes to 400 seeds. Okay, it depends on the size of the seed and size of the container of the substrate. Okay, and the test is conducted under favorable conditions of moisture, temperature, suitable substratum, which means medium. It can be of any type, soil, paper, or sand, and light. Okay, all the intensities, all the necessary raw materials are to be supplied in sufficient amount while testing the seed germination test. And no seed treatment is going to be given except those which are to be recommended by ISTA for seed germination test. Coming to the material required, what is substratum? It is a medium which serves as a moisture reservoir and provides a surface to the seeds, where the seeds can germinate and seedlings can grow. Okay, so it can be of 3 types: sand, pepper, and soil, especially when we are considering seed germination test. Okay, so the substratum can be taken as in these 3 forms. So let's discuss the first kind of substratum, which is sand. So what are the factors we have to look out for? The size of the sand particle. what kind of sand, what size is recommended. Seed particles should not be of too large or too small. It should be of optimum size. Okay, the sand particles should be of optimum size, and it should not be too large or too small, and it should pass through zero point 0.8 mm sieve and retain by 0.05 mm sieve. Okay, so it has to go through the sand that we are going to use as substratum is going to go through the sieves of different sizes, pore sizes. So it has to be stopped or retained by 0.05 mm, and it has to pass through 0.80 mm. Okay, so particular size seed sand, the size of the sand has to be selected. Okay, so this is for the easiness of germination and uh, growth growth and development of the reticle and plumule of the seeds. Okay, then coming to toxicity. Okay, sand should not have any toxic material or any pathogen or any nematodes in the sand. Okay, and if there is presence of any kinds of such, so it has to be sterilized with autoclave or any other methods. And next thing is germination tray. What kind of germination tray is required? It is usually used in the form of plastic trays. Although sometimes stainless steel is also used in some labs. And normal size of tray is 22.5 into 22.5 into 4 centimeter, which can capacitate 100 seeds at a time. Okay, so although smaller size trays can also be used. Coming to the method of seed placement, the method of sowing seed in the um, in the trays. Okay, uh, first one is seed in sand. What is seed in sand? Seeds are planted in a uniform layer of moist sand and then covered to a depth of 1 centimeter to 2 centimeter with a sand. So it is a two-step process. First of all, we have to uh, place the seed. Okay, place the seed into the sand, and it is to be covered by a layer of 1 centimeter or 2 centimeter of sand. Okay, coming to the second type, we have top of sand. In top of sand, seeds are pressed inside the surface with thumb or with with your fingers. Oh, and next is spacing requirements. So equal spacing on all sides has to be given to facilitate normal growth of seedlings and to avoid entangling of the roots while growing. And the spacing, the ratio of spacing should be in such a way that it should be 1:5 times the width or diameter of the seeds. Okay, so for so spacing will be a little bit higher for seeds such as rajma, while spacing would be a little bit lower for seeds such as brassica and rice grains. Okay, coming to water requirement, the amount of water to be added to the sand will depend on size of the seed. Okay, and for say for example for cereals except maize, okay, where water requirement is very high, the sand can be moistened to about 50% of its water holding capacity. Okay, and for large seeds such as legumes and maize, the moisture content should be 60% of the water holding capacity. Okay, coming to the second type of substratum, which is paper. Okay, and so for germination test, specifically for germination test, paper is the most widely used substratum because of its easy to find and low cost. Okay, and so this can, you can use any type of papers. Okay, well, it may be your filter paper and blotter paper or towel paper, paper towel that we usually use from day-to-day life. And it should have the capillary movement of water at the vertical direction, that is at the rate of 30 mm rise per minute. Okay, and it should be free from toxic substances and free from fungi and bacteria. Okay, So most of the this filter paper, okay, they came sterilized, so you can use directly, or they are not, if it is not contaminated, it's not of that much it usually is not contaminated by those pathogens which can infect the seed germination, so you can use directly. And it should hold sufficient amount of moisture during the period of test, okay, and things should be taken care of while germination test in paper. Things should be taken care. Water is not exhausted because the substratum can hold lesser amount of moisture compared to soil, okay. The texture should be such that the roots of germinating seeds will grow on and not in the paper, okay. Coming to the method of seed germination test in paper. First of all, top of paper. What is top of paper? Seeds are placed on one or more layers of uh, filter paper or blotter paper. Okay, so what we are going to do, we are going to cut the blotter paper or towel paper, or we can use directly the filter paper, and we are going to fold it in 2 to 3 layers. So why? Because we are going to make the folded papers retain more amount of water, okay. These paper plates are covered with lead and placed inside the germination cabinet, so it is easily assisted with a germination unit or a germinator, okay. Or sometimes if it depends on the temperature, you can directly grow in suitable room temperature, okay. And it is suitable for those seeds which require light, okay, because petri dish provides transparency in the and exposes it to the seeds, okay. And the next method is between paper method. The seeds are placed in between 2 layers of paper, and the third layer is a roll towel method, okay. When the seeds are placed between 2 layers of paper, and it is then rolled in towel. The rolled towel are placed in water source and kept in a germinator or germinating room. And next one is inclined plate method. So in case of inclined plate method, we are going to be using a glass plate, okay, upon which we are going to attach a germination paper attached with the seeds, okay. So, and we are going to incline it in at an angle of 45 degree from the ground, okay. Next thing is the third type of substratum, which is soil. And the type of soil that we have to choose for germination test is should be non-caking. It should be non-caking type, and it should not form. It should not have pebbles or large size of particles, stones or such. We have to remove. We have to grade the soil and go for seed germination test. Okay, and it must be free from weeds, seeds, bacteria, fungi, nematodes, and other toxic substances. Okay, so presence of which we can go for seed soil sterilization in autoclave again. Okay, and it is not recommended for reuse. So soil, when it is contaminated with the soil, if it is used for germination first, we have to dispose it. Okay, we don't have to use from time to time. And coming to the temperature requirement, after substratum we have temperature requirement. What is the required temperature? The soil, the substratum requirement, or the environment temperature requirement for germination, optimum temperature is 20 to 30. Okay, generally this may differ from crop to crop. Okay, and light requirement. So in light requirement, the light requirement of seed should be provided, especially for leafy vegetables, and it can also be provided for other type of crop plants. It depends on the type of crops. Okay, And so these are examples of some of the requirements of substratum, temperature, okay, and pre-treatment requirements. So you can see, so I'll take examples of peas between paper requirement or soil. Peas can be peas can use between paper and soil as a substratum for germination test. The temperature requirement is 20, and there is no requirement of pre-treatments recommended by ISTA. In case of brinjal, topo paper between paper substratum is usually recommended for seed germination test. A temperature of 20 to 30 degrees Celsius is recommended for germination test. Okay, and pre-treatment of ethereal ethiphone of 48 hours is needed. So these are some of the examples of requirements of during germination test. Okay, and it is recommended by ISTA. And the control of these temperature and temperature and light usually is done with the help of seed germinator, or else if the room temperature can provide a temperature of between 20 to 30 degrees Celsius, we can directly go for seed germination test in room temperature or in room. Okay, coming to next thing is seed apparatus. Okay, germinating apparatus. So the first kind of germinating apparatus is germinating cabinet, okay, which is called chamber, germination chamber, where in temperature and relative humidity is controlled. Okay, temperature, relative humidity, and light is also controlled here, and we can maintain the required temperature. So what we have discussed previously, the temperature here, and also the other requirements, the other conditions for favorable conditions for growth and development, such as light and relative humidity, can also be controlled in these chambers. Okay, next thing is a larger form of the germination chamber, which is known as room germinator. Okay, which is needed when you especially have to go for germination test of large, huge seed load. Okay, commercial scale. And this is a modified chamber of large one, and the worker can be entered into it and evaluate the seedlings. Okay, inside which you can do germination test for huge number of samples. Provisions are made to maintain the temperature and relative humidity inside the room. Okay, and this is widely used in practice, usually in seed companies. Okay, and next is counting board. It is an equipment. Okay, it is a small equipment, and it is used for counting the number of seeds. Okay, usually what is there, we have a plate which consists of two layers of steel plate. Okay, inside it, uh, so in this there is two layers of plate, one flat on the bottom, okay, which doesn't have any hole, and another one on the top which is having holes and such, and the number of holes is um, either 100 or 50. Okay, so whenever you take seeds, you pour seeds and you incline the seeds, all the uh, all the seeds which are not fitting in these holes will fall down, and seeds will retain in these holes. Okay, so you can every time when you do this, you can count 100 seeds at a time. Okay, so this is the basic idea of counting board. Counting of 100 seeds, if you have to count instantly or quickly, so you can directly uh, use this. Okay, so after taking the sample, the top plate is pulled in such a way that the holes are in one line, and that of the fixed number of seed falls on the substratum. Okay, yeah, this is the same only. Okay, Coming to vacuum counter, which is another equipment for seed counting. Okay, so in this it uses electric power to count the number of seeds. Okay, usually what is required here, a plate which consists of 50 to 100 holes. Okay, much like this. Okay, which is assisted into a pipe like this. Okay, and the with the use of vacuum, okay, with the use of vacuum, we are going to create pressure and absorb seeds. Although I haven't shown here, the plate is going to be attached here, and from the seed lot, with the use of vacuum pressure, okay, the the plate is going to absorb 50 seeds at a time or 100 seeds at a time. Okay, so in that case, in that way, the seed counts can be performed quite easily and fast. Next equipment is impression board. So what is impression board? It is a board in which we have protrusions or pins of 50 to 100. Okay, and well, it is useful in making holes into the tray or the seeds or the substratum where the seed is supposed to sow. Okay, and the knob lengths. Okay, knob lengths are arranged in equal lengths and space. Okay, and it gives, the usual purpose of this is to give impressions. Okay, to give holes on the sand to make it uniform and spacing for seeds. This usually saves effort and time for the seed germination test. And next thing is seed evaluation. Okay, after the germinating apparatus, we have seed evaluation. Okay, so according to ISTA, there are categories of seedlings which can be distinguished um, right after the germination test. Okay, so what are the type of seedlings that uh, is resulted from the germination test? We can classify based on the health, on the level of vigor and viability. Okay, first of all, we have normal seedling, abnormal seedling, and second one, abnormal seedling, and we have hard seeds, and then we have fresh ungerminated seeds, and the last one we have dead seeds. Let's see one by one. What is normal seed? So normal seedlings are those which shows capacity to continue development into normal plant when grown in favorable conditions of soil, water, and temperature. Okay, so in this case, length of the proper growth and development and length of the seedlings is also necessary, and uh, lack of decay and lack of disease infestations. Okay, and some of the characters which are uh, which a normal seedling should have, or it should have proper root, usually primary root, and this does not apply to crops such as rice because it is a fibrous root. So taproot system, so for those crops where taproot system is there, proper primary root is necessary. Okay, and well developed shoot axis, okay, consists of elongated hypocotyl in the seed, and epithelial germination is necessary. So proper root and shoot development should be there, and a well developed epicotyls in seedlings of hypogel germination. Okay, and one cotyledon in monocot and two cotyledons in dicot. Okay, so taking this as an example, two cotyledon development and one cotyledon development in monocot as well. So we have to check these criteria. A well-developed coleoptile in Gramine contains a green leaf. Okay, so in rice, okay, a protrusions of leaf from the coleoptile, okay, from the monocot seed is also one of the sign for normal seedlings. A well-developed plumule in dicots. And next thing is seedlings with following slight defects are also taken as normal seedlings. Okay, uh, with primary roots with limited damage but well-developed seminal roots systems in leguminous seeds, Gramini seeds, Cucurbitaceae, and Malvaceae seeds are taken as normal seeds. Seedlings with limited damage or decay to essential structures but not damage to the constructing tissues. Okay, so what is essential structures? The the cotyledon serves as a food material for growth and development of the growing plant. Okay, so if there is damage, there is decay or damage of small portion of the cotyledon. Okay, so it is not of much concern, but damage on the root or growing plumule and radicle will be a big maybe an issue on the normal seedling. Okay, and seedlings which are decayed by pathogens, but it is clearly evident that the parent seed is not the source of infections. Okay, these are also criteria for normal seedlings. And coming to the second one, which is abnormal seedlings. Okay, what are abnormal seedlings? Seedlings which do not show the capacity for continued development into normal plant when grown in favorable conditions of soil, water, and temperature. Okay, so deformity, all kinds of damaged seedlings, deformity seedlings, or some seedlings which are decayed are considered as abnormal seedlings. So it's classified as such. Okay, damaged, deformed, and decayed seedlings. Although uh, coming to the damaged seedlings part of abnormal seedlings, uh, what is this? Seedlings which with any one of the essential structure missing. Okay, maybe cotyledon, the whole cotyledon, or root, or maybe the plumule, or badly damaged seedlings, so that the balanced growth is not expected in the future. Exceptions to this is in case of germination in plant tissue culture. Okay, that is a different case there. In case of plant tissue culture, you can use growth regulators to initiate root or shoot. Okay, or germination, or any of them would work. But in case of when you're using substratum like soil, sand, or paper, you're not going to get protrusions of plumule, entire protrusions of radicle. Okay, and seedlings with no cotyledons, with splits and lesions or essential structures without primary root are considered as damaged seedlings. Okay, coming to deformed seedlings, with weak or unbalanced development of essential structures such as spirally twisted or stunted plumule, or hypocotyls or epicotyle, swollen shoot, stunted roots, etc. Okay, these usually, even though if they are successful in developing into a seedling, again they will grow to the proper size of the growth and development into a whole new plant. Okay, there will be stagnation in growth, there will be um, low in flowering. Okay, less number of flowering and less number of fruits. So it is better to remove such plants from them because it occupies space. Okay, because it is better to avoid these deformed seedlings, although these may develop into seedlings and plants. Decayed seedlings, which are especially infected by disease, okay, of any kind, bacteria, it may be bacteria, or it may be fungal. Coming to uh, another category of seedlings, which is hard seed, ungerminated, incapability of seed germination due to hard seeds. Okay, in case of hard seeds, example to be cited for are vegetable pulses, okay, or vegetable legumes. So in this case, there is inability to absorb moisture, especially it has to deal with moisture uh, inability to moisture, inability to absorb moisture either from the substratum or from the surrounding environment, okay, or the from the atmosphere, okay, till the end of the test period and remains hard, okay. So because it's usually because of the hard seed core which covers it. Once the seed cover is cracked, okay, so seed treatment in these kind of hard seeds are usually done, and the seed germination can be recovered in this scarification, a process called scarification, which means rubbing of the seed coat or cracking of the seed coat so that moisture, a little amount of moisture enters inside the seed coat, and the usual process, usual metabolism of growth and development starts, okay, right after the moisture entry. And example of this again is legume seeds. And next is fresh ungerminated seeds, seeds which looks usually fresh but lacks viability, vigor, okay. Next is dead seeds. Seeds at the end of the test period are neither hard nor fresh or have produced any part of the seedling. Often dead seed collapse and milky paste comes out when pressed at the end of the test. Okay, some of the seeds may look healthy, and some of these seeds usually when undergo for seed germination test, they will not germinate at all, and at the end of it, it will result in formation of milky paste or dead seeds, okay, and it will result to no germination, and better to avoid such seeds, okay. So retesting is necessary, okay, if the germination test is not sufficient or not satisfactory. If the result of the test is considered unsatisfactory, it should not be reported, okay, and a second test shall be conducted, or by the same method or by alternative method under the following circumstances, okay. So under these conditions, retesting is going to be done. Replicate performance is out of tolerance, okay. That means when you're going to do germination test in 4, okay, 100 in 4, that we have like the one we have discussed in principles of seed germination, 100 seeds in 4, which makes 400. So if one of the replicate, okay, 100 seed replicate in a tray is out of tolerance, that means it doesn't show the uh, level of germination like others, okay. So we should repeat the that replicate, okay. It results being inaccurate due to wrong evaluating of seedlings or counting or errors in test conditions, okay. So inaccuracy in evaluating the seedlings, okay. The error might happen, and errors might happen at any time. And dormancy, persistence, or phytotoxicity of fungal or spread of fungi or bacteria, okay. When unexpected fungi and bacteria intrudes into the seed germination test, so we also have to take care of this, reconsidering the seed germination test. What is the use of tolerance? The result of germination test can be relied upon only if the difference between the highest and lowest replicate is within acceptable tolerance, okay. Like we have discussed, 4 replicates of 100, okay. So if the one of the replicate is showing 5 normal seedlings, and one of the replicate is showing um, 90 normal seedlings, okay, there is a huge huge difference in the uh, record, okay. So it should not be that much different. The difference should not be at that level. Although the difference can be there, that's why replication is done. The replication should not be at that range, okay. And which is called tolerance, Okay, if it surpasses the tolerance, we have to replicate, or we have to repeat the test. um, to decline, if two test results of the same sample are compatible, again the tolerance table is used. So that tolerance table is here, okay, which where the amount of where the difference between the highest and lowest amount of germination and tolerance level is written here. Okay, so we have to go according to the germination test, has to go according to that. Okay, if it fulfills, we can move forward. If the germination test doesn't fulfill the tolerance, so we have to repeat the uh, germination test. Coming to reporting results, okay, the results of germination test is calculated as the average of 4 into 100 seed replicates, 4 replicates of 100 seeds. Okay, it is expressed as a percentage of number of normal seedlings. Okay, although we have here the formula is given here, and the percentage is calculated to the nearest whole number, not in fractions. Okay, and percentage of abnormal seeds, hard seeds, fresh and dead seeds is calculated in the same way. Okay, so that to analyze the amount of deformity abnormal seedlings, okay, these should be entered in the analysis of certificate under appropriate space. Okay, if result is nil for any of these categories, it should be reported as 0. Okay, if any of these is not there, if there is 100% normal seeds, for all the columns of these abnormal seedlings, hard seedlings, fresh ungerminated seeds should be written as 0. Okay, taking these as an example, we have here a germination register book. Okay, so in this we are going to record the name of the grower, seed germination tester, and the accession number, crop code, okay, the type of crop, and moisture content. In germination test, germination percentage is going to be there, number of dead seeds. Again, although spelling is incorrect here, DEAD, dead seeds, hard seeds, abnormal seeds, normal seedlings. Okay, and such and such data has to be recorded precisely in the entry. Okay, so coming to the end of the slide, end of the class, if you have any questions you can ask.